Chalo bachcho, let's start. Have you ever really thought about it? How is this incredibly complex machine, your body, even built? Well, it all comes from one single blueprint, your DNA. But what happens if there's a small printing mistake in that blueprint? A tiny typo? Today, we're going to explore exactly that. These tiny errors in our genetic code that can sometimes lead to very, very big diseases.
Just imagine, your DNA is like the ultimate instruction manual. I mean, the most detailed one you can ever think of. Everything, from your eye color to how your heart beats, every single instruction needed to build you and run you is in there. So the question is, what happens if there's just one little mistake in that manual? Even a tiny one. So that typo we're talking about, it has a proper scientific name. We call it a mutation. Simple as that. It's basically a permanent change in your DNA sequence. This is the typo in our blueprints. And this one word, mutation, is the starting point for this entire chapter, for everything we are going to discuss today. Okay, let's dive into the details now. You see, not all typos are the same, nah? Some are very small, just like changing one letter in a word. But others can jumble up the whole sentence so badly that it makes no sense at all. It becomes completely unreadable. So let's break down the main type. Broadly speaking, there are two big categories you absolutely need to remember. First, you've got point mutations, and second, frame shift mutations. Think of a point mutation as just swapping one letter for another in a word. It seems small, but trust me, it can be a total game changer. But a frame shift mutation? Arembapere, that's much more dramatic. It's like adding or deleting a letter, and poof, the entire rest of the message gets completely scrambled. So you might be thinking, sir, how bad can one tiny letter change really be? Well, let me tell you. Take beta thalassemia, a very serious blood disorder. In this condition, just one single base change, just one, transforms a regular instruction into a sudden stop sign. It's like telling the factory to just stop production right in the middle of the assembly line. The machinery just halts. The protein is never finished. And this, my friends, is what we call a nonsense mutation. Now, if you thought that was bad, frame shift mutations are pure chaos, total chaos. Imagine our genetic code is read like a sentence in groups of 3 letters, like the cat ate the rat. A frame shift is when someone just adds or removes one random letter. Suddenly, the whole reading frame shifts. The sentence becomes something like THFE-cat-tat. It's complete gibberish. And the protein that gets made from this nonsense, you guessed it, completely useless. So we've understood the typos, but how do they actually translate into diseases in a real person in the clinic? Well, it turns out the scale of the glitch, the size of the mistake, that matters. And it matters a lot. We can classify these genetic disorders into 3 main buckets, based on the scale of the problem. First, we have monogenic disorders. Mono means one, right? So this is caused by a big mistake in just one single gene. Next, we have the complex or multigenic disorders. These are like a perfect storm, you know? Many small genetic glitches plus environmental factors all coming together. And finally, you have the big ones, chromosomal disorders. Here, the error is massive. It's like a whole chapter of the instruction manual is either missing or it's been copied twice. For today's class, we are going to zoom in on that first category only, monogenic or single-gene disorders. These are really fascinating because a huge life-altering problem can be traced back to just one single faulty gene. And what's more, they often pass through families in very clear, predictable patterns. Okay, chalo, section 3. So now the question is, how are these single-gene typos actually inherited? How do they get passed from parents to children? You'll see the patterns are actually surprisingly logical. The 2 patterns you'll see most often are autosomal dominant and autosomal recessive. In dominant conditions, it's simple. You only need one bad copy of the gene from just one parent, and that's enough to cause the disease. But for recessive conditions, you need to be a bit unlucky. You have to inherit 2 bad copies, one from your mother and one from your father. And often, the parents are just carriers, completely healthy, with no idea they're even carrying this faulty gene. And then there's a very interesting twist in the story, the X-linked disorders. As the name suggests, here the faulty gene is located on the X chromosome. Now you remember from your basics, na? Males have one X and one Y chromosome. Females have 2 X chromosomes. So because males have only one X, if that one X has the faulty gene, that's it. They will have the disease. That's why conditions like hemophilia are seen so much more often in boys. All right, section 4, from code to condition. Let's connect all this theory to what you will actually see in the clinic. How does a small typo in the genetic code cause real physical symptoms? Let's take two fantastic examples, both involving structural proteins, the proteins that literally act as the steel and cement of our bodies. First case study, Marfan syndrome. This is a classic example. It's a disorder of the body's connective tissue. And what is connective tissue? Well, it's exactly what it sounds like. It's the biological glue, the cement that holds everything together. All our cells, our organs, everything.
Now, patients with Marfan syndrome, you can often spot them. They have a very distinct look. They're usually unusually tall with very long arms, long legs, and long, thin fingers. But the really dangerous stuff, the life-threatening part, is what you can't see. It's internal. Their aorta, the mini artery coming from the heart, becomes weak. It can stretch out like a balloon, an aneurysm, and can even tear, which is a massive medical emergency.
And what's so incredible about this is that all these problems in the skeleton, in the eyes, in the heart, all of it comes from a defect in just one single gene. That gene is FBN1. Its job is to make a protein called fibrillin-1. And fibrillin-1 is absolutely essential for our connective tissue. It gives it strength and elasticity.
Now listen to this. This is the really interesting part about its pathogenesis. The faulty fibrillin causes a two-pronged attack. It's a double-hit problem. First, the obvious one. Because fibrillin is abnormal, the connective tissue loses its structural support and becomes weak. But second, and this is key, it also releases too much of a growth factor called TGF-beta, which it's normally supposed to hold on to. This combination, weak tissue and excessive growth signal, is what causes the whole clinical picture. It's like a 1-2 punch from a single genetic type.
Okay, moving on to our second case study. This isn't just one disease, actually. It's a whole group of related disorders, which we call the Ehlers-Danlos syndromes, or EDS for short. The common link, the one thing that connects all types of EDS, is a defect in collagen. You all know collagen, right? It's the most abundant protein in our body, the main structural protein. So when your collagen is faulty, you get these classic signs. Skin that is super stretchy, joints that are hypermobile, they can bend in ways they're not supposed to, and very fragile tissues that can bruise or tear very easily. An EDS is a brilliant example of something we call genetic heterogeneity. What does that mean? It means the same clinical picture can be caused by mutations in many different genes. Think of it like a car factory. The final car has a defect. Why? Maybe the gene for the steel was bad, or maybe the gene for the enzyme that puts the screws in was bad. Different problems in the assembly line, but the end result is the same, a faulty car. Same thing here.
So after going through all of this, all these details, what is the main takeaway point? What is the one thing I want you all to remember from this? Remember this, there are 3 major categories: single gene, multigenic, and chromosomal. It all comes down to the scale of the problem. A genetic disease is not a one-size-fits-all thing. The problem can be a tiny error in one gene, a complex mix of many genes plus your lifestyle, or a huge, massive error involving a whole chromosome. So ultimately, understanding genetic disease is really about understanding scale, the size of the typo in your body's blueprint, whether it's just one single wrong letter or an entire missing chapter. That is what determines the nature and the story of the disease. Look, our genetic blueprint tells the story of our past, of our evolution, and understanding it is absolutely fundamental to modern medicine. But it also makes you think, nah? It leaves us with a very big question for the future. If this code tells the story of where we came from, how is our new ability to read and even edit this code? How is that going to shape the story of our future? Namaste, students. Welcome back. Today, we're going on a fascinating journey. Not to a hospital ward, no. We're going much, much deeper, right inside the cell, to meet a tiny but mighty hero who acts as our cellular housekeeper. And we'll also find out what happens, what absolute chaos unfolds when this little hero decides to go on strike. Just think about it. Inside every single one of your cells, there's a microscopic recycling plant. It's working 24/7, non-stop. Now, this little plant is absolutely key to keeping the cell healthy and clean. But what happens when it breaks down? Well, the garbage just starts piling up, and the consequences, believe me, they can be absolutely devastating.
Okay, Bacho, so here's our game plan for today. First, we're going to properly meet the cellular housekeeper at the lysosome. Then, we'll look at the mess that happens when the system breaks. After that, we'll get into two classic case files you must know for your exams, Tay-Sachs and Gaucher disease. And don't worry, we'll finish on a high note, looking at some of the hope on the horizon. Ready? Let's begin. Right, Section 1, the lysosome, our cell's housekeeper. The best way to think of the lysosome, it's like the cell's very own kabadiwala. You know, the recycling guy. It's this tiny little organelle with a massive job, breaking down all the waste, the old machine parts, all the junk the cell doesn't need anymore. Now, don't just think these are simple bags of acid, okay? No, they are super specialized compartments packed with powerful enzymes. And their job involves two key processes. One is autophagy, that's self-eating, where they gobble up the cell's own old parts, like worn-out mitochondria. The other is heterophagy, where they digest stuff that comes from outside the cell. It's basically the ultimate quality control system. Okay, so on to Section 2. What happens when this system breaks? Imagine that recycling plant has an assembly line of workers, each one a specific enzyme. Now, what happens if just one of those workers doesn't show up for their shift? Chaos. The whole assembly line stops. Everything just piles up. And that's the tragedy here. It's often just a single genetic mistake, one missing enzyme, that brings the whole system to a grinding halt. See, in a healthy, normal cell, the process is beautiful. It's seamless. Big, complex molecules come in, and the enzymes, let's call them worker A, worker B, and worker C, they work in a perfect sequence. They break everything down into small, usable parts that the cell can recycle. It's a super clean, super efficient system, no problem. But now, look at this. It's a disaster. Here, our enzyme B, our worker B, is missing, and the assembly line is stuck. The process stops, dead in its tracks. All this undigested material just builds up and up and up. This is what we call primary accumulation. The lysosome gets bloated, it swells up, and it starts messing with the entire cell's function. And it gets worse. This also leads to a huge secondary problem, defective autophagy. The old, damaged mitochondria don't get cleared out, so they start leaking toxic free radicals. And this ultimately can kill the cell. All right, Section 3. Let's get into our first case file, Tay-Sachs disease. And I have to tell you, this is a truly tragic, heartbreaking condition. It's a devastating neurological disorder that hits infants. And it's particularly common in certain populations, like Ashkenazi Jews, where the carrier rate is shockingly high. Something like 1 in every 30 people. So what's the core problem here? Pay attention. The missing enzyme is hexaseminidase, A. Without this enzyme, a specific kind of fat, a lipid called GM2 ganglioside, just can't be broken down. Now, these gangliosides are actually really important for the brain to work properly. But when they pile up inside the neurons, they become toxic. They literally poison and destroy the nerve cells from the inside out. Now, pay close attention, Bacho. This is a classic for your exams. Look here. On the left, what you're seeing is a neuron that's completely ballooned out. It's swollen with these vacuoles. Those vacuoles are the lysosomes just stuffed to the breaking point with lipids. And on the right, under the electron microscope, you see the telltale sign, these weird, whorled, onion skin-like layers inside the lysosomes. You must remember this image. These are the diagnostic hallmarks of Tay-Sachs. Clinically, the story is just heartbreaking. A baby is born, seems perfectly healthy, and then, around 6 months of age, things start to go wrong. There's this relentless decline, motor skills, mental function, everything goes downhill. They eventually become blind, their muscles go limp, and they develop this very characteristic cherry red spot in the macula of the eye. Now, it's not totally unique to Tay-Sachs, but it's a huge clinical sign. And sadly, these children end up in a vegetative state and usually pass away by the age of 2 or 3. Okay, let's move on to our next case file, Gaucher disease. Now, this one is actually the most common of all the lysosomal storage disorders, but you'll see its clinical picture is very, very different from what we just saw with Tay-Sachs. So in Gaucher disease, the culprit enzyme is different. It's glucocerebrosidase. And the garbage that piles up is another lipid called glucocerebroside. But here's the key difference. Listen carefully. The accumulation doesn't happen mainly in neurons. Instead, it happens in the big eating cells of the body, the phagocytes. You know, the macrophages. And you find those all over the place, in the spleen, the liver, the bone marrow. And this, my friends, is another picture you have to burn into your memory.
This is the famous Gaucher cell. It's a massive macrophage, sometimes growing up to 100 micrometers. It's huge, and it's just packed with these lipids. And look at its classic appearance. It's beautiful in a pathological way. The cytoplasm doesn't look foamy like you might expect. No, it looks fibrillary. It has this classic crumpled tissue paper appearance. Why? Because the stored lipid is arranged in these little stacks. Unforgettable. Now, Gaucher's isn't just one disease. There are different types. Type 1 is the most common, and luckily it doesn't affect the brain. It causes a massively enlarged spleen and liver and serious bone problems. But types 2 and 3 are much more severe, and they do involve the brain. But here is the most incredible, surprising connection, and this is clinically very important. Just being a carrier for Gaucher disease, not even having the full disease, is the single biggest known genetic risk factor for developing Parkinson's disease. Isn't that amazing? The thinking is, it's that same problem, impaired autophagy. The cell's garbage disposal is slow, so other toxic proteins, like the alpha-synuclein you see in Parkinson's, also start to build up. Okay, our final section, and after all this doom and gloom, I want to end on a hopeful note. Because while these diseases are really terrible, modern science is fighting back. And it's fighting back with some really, really clever strategies. We basically have 3 main approaches to treatment now. The first one is pretty straightforward: enzyme replacement therapy. You just give the patient the enzyme they're missing, usually through an IV drip. Simple. The second is called substrate reduction therapy. The logic here is also simple. If your garbage disposal is slow, well, just try to make less garbage. And the third one, this is really clever, is molecular chaperone therapy. See, in many of these cases, the body is actually making the enzyme, it's just misfolded and doesn't work right. So we can give these tiny molecules that act like a chaperone to help the enzyme fold correctly, so it starts working again. This is really a new era for treating genetic diseases. So let's end with this thought. These rare and tragic genetic mistakes, they've actually been an amazing window into the cell. They've taught us so much about fundamental biology, like just how critical autophagy is. You know, this link between Gaucher and Parkinson's, that's just a tip of the iceberg. So the big question we're left with is this: what will these rare diseases teach us next about the much more common diseases that plague us, like Parkinson's and Alzheimer's? That's what makes this field so exciting and so incredibly important. Thank you. All right, everyone, settle down, settle down. Welcome back. So, we're continuing with Chapter 5. Last time, we talked about the basics of our genetic code. Today, we're diving into the really fascinating and clinically very important world of what happens when that code goes wrong. We're going to look at everything from a single faulty protein to entire missing blueprints. Okay, bachcho, look here. This is our plan for today. We have 5 main topics to get through. Now, I want you to pay very close attention, huh? Everything you see on this slide is extremely high yield for your exams. So, notebooks out, let's begin. Chalo, let's start with a simple idea, right? Inside the cell, inside the cytoplasm, we're going to talk about how the body manages its fuel. Glycogenesis. I know. It's a big, scary word. Don't worry about it. It sounds very complicated, but it's really just a fancy term for a simple, logical problem. The body has stored its energy, its sugar, but it can't get to it when it needs it most. That's it. Think of it like a bank account for your body's energy. It's a simple 2-step process. First, after you eat a meal, you deposit extra glucose as glycogen. That's money in the bank. Later, when you need that energy, your body makes a withdrawal, breaking glycogen back down to glucose. Simple, right? But what happens if the ATM, the enzyme, is broken? Now this table, this is brilliant. Why? Because it shows us a fundamental principle of pathology. A different broken enzyme leads to completely different problems. Look here. In von Gehrig disease, the liver's enzyme is kaput, so the patient gets severe hypoglycemia. In myocardial disease, the problem is in the muscles, so patients get these terrible cramps during exercise. And in Pompe disease, the problem is everywhere, which leads to a massively enlarged heart. Do you see the pattern? The location of the broken enzyme dictates the entire clinical picture. It's beautiful. Logical pathology. Okay, class, everyone, eyes here. This is where it gets really exciting, you know? This is pathology you can actually see under the microscope. On the left, you've got healthy heart muscle, nice and pink. On the right, a heart from a patient with Pompe disease. You see all those white, empty-looking spaces inside the cells? That is glycogen just piling up, suffocating the cell because it can't be used. This is what we mean by massive cardiomegaly at a cellular level. It's remarkable, isn't it? Okay, so we've just talked about a broken part. A hardware problem, right? An enzyme? Now let's move up a level. Let's talk about a problem with the instructions themselves. Think of it less as a hardware problem and more like a software bug in the DNA code.
You see, the DNA sequence, the genetic code itself, that's the hardware. But you need software to run it, correct? That is epigenetics. It's this beautiful, complex system of tags and switches that tells our genes when to turn on and when to be silent. So in Rett syndrome, a crucial silencing protein called MECP2 is faulty. Now, just imagine this scene. A young girl developing perfectly normally for the first year or so, hitting all her milestones. But then, this critical switch fails. Genes that are supposed to be silent are not turned off. And the result is a devastating neurodevelopmental regression. She loses her speech, her motor skills, and develops these classic purposeless hand-wringing movements. It's a heartbreaking example of what happens when the software goes wrong. All right, so we've gone from a single protein to a single gene's instructions. Now, let's zoom out. Let's zoom way out to the big picture. We're going to look at the entire genetic blueprint, the chromosomes themselves. What you're looking at here, this is it, our complete genetic blueprint, a karyotype. 22 pairs of autosomes and the 2 sex chromosomes, all neatly arranged by size. Think of it like a 46-volume encyclopedia for how to build a human. Any error in this map, a missing volume, an extra copy, can have major, major consequences. So the most basic, the most fundamental error is just having the wrong number of chromosomes. It's that simple. The correct balanced set of 46 is called euploidy. Anything else is aneuploidy. If one is missing, it's called monosomy, mono for one. If there's an extra one, it's called trisomy, tri for three. Remember these terms, they're absolutely crucial. Now, besides having the wrong number of books, the books themselves can have, well, printing errors. These are what we call structural mistakes. Think of it this way. In a translocation, a paragraph is cut from one chapter and pasted into another one. In a deletion, a whole sentence is just gone. And in an inversion, a page has been bound upside down. See? Simple analogies for complex errors. Okay, with that background, now you understand what can go wrong. So let's look at the most common chromosomal disorder you will all see in your clinical practice. For sure. 1 in 700. Remember this number. Write it down. That is the incidence of Down syndrome. And in the vast majority of cases, about 95%, it's caused by that simple numerical error we just talked about, an extra copy of chromosome 21, trisomy 21. Okay, now imagine you are the doctor. The lab sends you this report. This amazing technique is called FISH. The green dots are our control. We expect to see 2 of them. Now look at the red dots, which are the probes for chromosome 21. Count them with me. 1, 2, 3. 3 red signals. What does that mean? 3 copies of chromosome 21. Diagnosis confirmed. See? This is the power of modern cytogenetics right here. So what does one tiny extra chromosome actually do? Well, it all comes down to something we call the gene dosage effect. It's a simple idea, really. Our bodies are designed for 2 copies of most genes. Having 3 copies, well, it's like having a recipe that says 2 cups of sugar, but you put in 3. The cake will still come out, but it will be fundamentally different, right? That overdose of genes leads to this classic combination of features you must know: the flat facial profile, intellectual disability, the high risk of heart defects, and that tragically early onset of Alzheimer's disease. All right, finally, let's talk about aneuploidies of the sex chromosomes. And what's really fascinating here biologically is that having an extra or missing X or Y is, well, it's surprisingly well tolerated compared to errors in the other chromosomes, the autosomes. We're going to focus on the 2 most important syndromes, the ones you absolutely 100% must know for your exams. On one side, we have Klinefelter syndrome in males, caused by an extra X chromosome. And on the other side, we have Turner syndrome in females, where one X chromosome is completely missing. For Klinefelter, the key thing to remember is XXY. Simple, an extra X chromosome in a male. This leads to what we call hypogonadism, meaning underdeveloped testes, and a very distinctive body shape, with unusually long legs. And a key clinical point, because this testicular failure only becomes obvious at puberty, it's often not diagnosed until the teenage years. And for Turner syndrome, you have to remember monosomy X, just one X chromosome in a female. The classic features you'll see are short stature and what we call streak ovaries. The ovaries fail to develop, and this leads to infertility. You'll also often see a webbed neck and a high risk for specific heart defects, especially coarctation of the aorta. So, as we've seen today, a single faulty enzyme, a misplaced epigenetic switch, one extra chromosome, these seemingly small errors can change a person's entire life. But here's the thing to remember, it's not random chaos. It all follows clear, logical genetic rules. And as you go on to become doctors, I want you to always appreciate that delicate, incredible, and sometimes fragile balance required for normal human development. That's all for today. Thank you, class. All right, class, settle down, settle down. So you all remember Gregor Mendel from your textbooks, right? Dominant, recessive, all that straightforward stuff. But let me tell you, when you're actually in the clinic, the real fun begins when genetics decides to break those rules. And that's exactly what we're going to talk about today. The fascinating exceptions, the real puzzles that show us just how incredibly complex our DNA really is. Chalo, let's get started. So just imagine this for a second. You're in your rotation, looking at a family's pedigree chart. And the pattern, it just makes no sense at all. It's not dominant, not recessive, definitely not X-linked. You're scratching your head thinking, what on earth is going on here? Well, that's not a mistake in the chart. No, it's a clue. It's telling you that a much deeper, much more interesting genetic story is playing out. Okay, first up, let's talk about what I call the stuttering gene. Imagine a tiny part of the DNA code that starts repeating itself, kind of like a stutter. Now, with each new generation, it's like a faulty photocopier that just keeps making more and more copies of that stutter until it becomes a massive, massive problem. This is the world of trinucleotide repeat disorder. Now, the classic example, and listen, I want you all to put a big star next to this in your notes for your exams, is Fragile X syndrome. It is the most common hereditary cause of intellectual disability in males. And the culprit? It's a huge expansion of a three-letter DNA repeat, CGG, in one single gene called FMR1. So how does this stutter get worse? Just look at these numbers here. Normally, a person would have somewhere between 6 and 55 of these CGG repeats. That's fine. But some people carry what we call a premutation. That's between 55 and 200 repeats. Usually, they're okay. The real problem starts when a woman with this premutation has children. That repeat, it can just explode, massively expand into a full mutation with over 200, sometimes even thousands of repeats. And this phenomenon, where the disease gets more and more severe, or it shows up at an earlier age in the next generation, it has a specific name. You must know this. It's called anticipation. What's happening here is that the gene repeat is really unstable. It expands dramatically when it's passed down from the mother during the formation of her eggs. And this is why you can see a grandfather who only has a mild premutation, but his grandsons have the full-blown severe syndrome. So what does this huge repeat actually do? Well, think of it like a giant sticky piece of chewing gum that's just gumming up the works of the gene. The cell sees this big mess and it panics. It just shuts the entire gene down using a chemical off switch we call methylation. This completely stops the production of a really vital brain protein, FMRP, and it's the absence of this protein that causes all the features of the syndrome. All right, moving on to case number 2. Now, for this next group of diseases, you can completely ignore the father's genes. Seriously. Because for this small but incredibly powerful part of your DNA, you only have your mother to thank. You have to remember, you actually have 2 genomes inside you. There is the famous one, of course, in your cell's nucleus with all the DNA from both your mom and your dad, but you also have this tiny separate genome tucked away inside your mitochondria, the powerhouses of the cell. And that mitochondrial DNA, it comes exclusively from your mother's egg cell. A textbook example of a mitochondrial disease is Leber hereditary optic neuropathy, or LHON. It's a really devastating condition. You see young adults, mostly males, and they experience this rapid, progressive blindness. The problem isn't actually in the eyes themselves, but in the optic nerve cells. They're dying off because their mitochondria just can't produce enough energy. Now, look at this pedigree chart. I'm telling you, this is the most striking pattern you will ever see in genetics. Look closely. If the mother is affected, all of her children, boys and girls, doesn't matter, they all get the condition. But if the father is affected, he passes it to absolutely none of them. 0. If you ever see this pattern in a clinic or on an exam, your first thought must be mitochondrial inheritance. It's a dead giveaway. All right, let's get to our final case, and this one is perhaps the most mind-bending of all. What if you have 2 perfectly normal copies of a gene, one from each parent, but the cell just decides to listen to only one of them? This is the very strange world of genomic imprinting, where some genes come with a do not disturb tag already attached from one of your parents. The whole fun to hear is that for certain genes in your body, only one copy, either the one from your mother or the one from your father, is actually active. The other copy is chemically silenced or imprinted. Now, this is not a mutation in the DNA sequence itself. It's more like an instruction written on top of the DNA, like a little sticky note telling the cell, hey, read this copy, not that one. And here is where it gets truly wild. A tiny little deletion in one specific region of chromosome 15 can cause two completely different, totally distinct clinical syndromes, and the only thing that matters is whether you inherited that deletion from your father or from your mother. So here's what's happening on chromosome 15. In this one particular region, you have some genes that are only active on the copy you get from your dad, and then you have another gene that's only active on the copy from your mom. So you can see if that paternal copy gets deleted, you lose its function and you get one disease. But if the maternal copy gets deleted, you lose its function and you get a completely different disease. So a deletion from the father's side results in Prader-Willi syndrome, which is characterized by this constant, insatiable hunger. But a deletion on the mother's side, of the exact same region, mind you, causes Angelman syndrome, with its characteristic happy puppet kind of walk and what we call inappropriate laughter. Isn't that just incredible? Two completely opposite syndromes from one single genetic address. The only difference is which parent it came from. So I know what you might be thinking. Professor, these are all rare diseases. Why should we focus on them so much? You should care because these exceptions, they don't just break the rules. They actually reveal the deeper rules of how our genome works. For you, as a future clinician, recognizing these patterns isn't just trivia, it's absolutely essential. So let's just quickly recap the main points. First, the stuttering gene, those trinucleotide repeats that expand and cause anticipation. Second, the mother's legacy. Mitochondrial DNA is passed down from a mother to all her children, period. And third, the silence gene, genomic imprinting, which means for some genes, it's not what you have, but who you got it from that really counts. You see, understanding these non-Mendelian patterns is absolutely critical for you. When you're sitting with a family and they have a complex history, being able to recognize these clues, that can lead you straight to the right diagnosis, and that is always the first step in providing accurate genetic counseling, managing the patient, and helping the entire family understand what's happening. The genome is just so much more complex and dynamic than Mendel ever could have imagined. These exceptions we talked about today, they were once complete mysteries. It really makes you wonder, as our technology for reading DNA gets even more and more powerful, what other fundamental rules of genetics are we just on the verge of discovering? Think about it. We'll see you in the next class.
All right, everyone, settle down, settle down. Let's begin. So, today we're going to do something a little different. We're going to become detectives, but our crime scene, it's the human body, and the mystery is hidden in the most complicated code you can imagine, our DNA. We're going to learn how to crack the code of genetic diseases. So think of this as a special investigation. Let's get started.
Now, just to give you an idea of the challenge we're up against, think about this. Your entire genetic code, your genome, has about 3 billion letters. A single devastating genetic disease can be caused by just one of those letters being wrong. One. That's like trying to find a single spelling mistake in a massive library filled with thousands and thousands of books. I mean, where do you even begin? How is that possible? Well, it turns out our detective kit has some pretty amazing tools.
So our first tool is for finding the most obvious clues. We're not looking for a single wrong letter here. We're looking for a whole missing chapter, or maybe an extra chapter that's been copied and pasted in by mistake. For these big, large-scale changes, we use a technique called FISH.
So what is this FISH, eh? The idea behind it is actually quite brilliant. Imagine you have a special key that glows in the dark, and this key is designed to fit only one specific lock in the entire world. In genetics, that glowing key is our fluorescent probe. It's a tiny piece of DNA that we designed so it will stick to only one specific part of one chromosome. And when it sticks, it lights up, showing us exactly where that piece of the code is.
Okay, so when do we actually use this glowing key? Well, you use it when you already have a strong suspect in mind. For example, if a child has symptoms of Down syndrome, we can use a probe that's specific to chromosome 21. If we look under the microscope and see 3 glowing spots instead of the usual 2, our suspicion is confirmed. Simple. In cancer, we use it to see if a particular gene has been copied too many times, what we call amplified. And one of the best things about this test is its speed. In really urgent cases, a fast diagnosis can make all the difference.
But what happens when the detective has no suspects? You can't use a special key if you don't know which lock to check, right? In that situation, you need to cast a much wider net. You need to scan the entire crime scene for anything that looks out of place. And that is where our next tool comes in, genomic arrays. You know, this technology has improved so much over the years. The older version, Array CGH, was a bit basic. It would compare a patient's DNA to a normal sample and could tell you if any big pieces are missing or duplicated. But the modern SNP array, oh, that's a whole different level. It not only sees those big changes, but it also checks for thousands of single-letter variations across the whole genome. This gives us such a high-resolution view that we can spot really subtle clues. Things like uniparental disomy, where a child gets both copies of a chromosome from just one parent. That's something the older tech would completely miss.
And here, have a look at this. This is a real-world example of just how powerful this is. This data is from a young patient with leukemia. Let me walk you through it. The top part of the graph measures the amount of DNA. Now notice this area here. See how the dots suddenly drop down? That's our first big clue. It means a piece of chromosome 12 is missing. Now look at the bottom graph. This part shows all the genetic variations. In the normal areas, you see 3 clear bands of dots. But look what happens in that exact same section where the copy number dropped. Poof, the middle band is completely gone. This confirms it. One copy of that part of the chromosome is lost. It's like a high-definition map that pinpoints the problem for us.
Okay, let's switch gears a bit. What if the bad gene, our culprit, is a master of disguise? We don't know what it looks like, we don't know where it's hiding. What do we do then? Well, we have to change our strategy. Instead of looking for the culprit directly, we start tracking its known associates, its friends, as they move through a family. The principle here is called genetic linkage, and it's actually quite simple. Think of it this way. Genes that are physically very close to each other on a chromosome, they're like two friends holding hands. When the DNA gets passed down from parent to child, they almost always get inherited together. So, if we can find an easy-to-spot genetic marker that's always hanging around with our unknown disease gene, we can just use that marker as a tag to track the real culprit through the family's generation. A classic example of this is polycystic kidney disease, PKD. For a very long time, scientists just could not find the gene that caused it. So they tried something different. They found a nearby DNA marker that had different lengths in the family. Let's just call the versions A, B, and C. Then they mapped out the family tree and saw who had the disease. And a crystal clear pattern emerged. Every single person with PKD had also inherited marker C. That was the breakthrough. It meant the disease gene and marker C were linked. They were holding hands. From then on, they could test young family members just for marker C to see if they were at risk. Really, a very clever piece of detective work.
All of these tools are fantastic, but they're still about finding clues and hints. But now we come to the ultimate weapon in our detective's toolkit. This is the technology that lets us stop searching for clues and instead just read the entire instruction book from cover to cover, letter by letter. This is the revolution that we call next-generation sequencing.
So what makes NGS such a massive game changer? It's this idea of being massively parallel. You see, the old way of sequencing was like reading a very long book, one word at a time. So slow. NGS is like having millions of tiny machines reading millions of different parts of the book all at the same time. This incredible parallelism is what makes it so unbelievably fast and so much cheaper than it used to be. It has completely changed genetics.
The process itself is just ingenious. First, you take the DNA and break it into millions of tiny little pieces. These pieces are then stuck onto a special slide, each one in its own little spot. Then, in step 2, each piece is copied over and over again to amplify the signal, make it easier to see. And then the sequencing starts. The machine adds the DNA building blocks A, C, G, and T one by one, and each letter has a different colored fluorescent tag. After each letter is added, a camera takes a picture, recording the color at every single spot on the slide. By stacking all these pictures, the computer can read the sequence of colors for each tiny fragment and piece together millions of DNA sequences all at once. It's just amazing.
And we can use this powerful technology at different levels of focus, kind of like a camera with a zoom lens. For a very specific investigation, we can use a targeted panel to look only at a handful of known suspect genes. If we need a wider view, we can use whole exome sequencing to read all the protein coding regions of the DNA.
And for the most complete investigation possible, the whole shebang, we can use whole genome sequencing to read the entire 3 billion letter code.
This brings us to the future of our detective work. We are now at a remarkable new frontier. The ability to uncover the secrets of a hidden tumor, not by doing a painful surgery to get a biopsy, but by analyzing a simple tube of blood.
This is the concept of the liquid biopsy. You see, tumors aren't just sitting there quietly. They're constantly shedding tiny fragments of themselves into the bloodstream. This can be whole cells, bits of DNA, or other molecules. A liquid biopsy is a revolutionary test that allows us to capture and analyze these microscopic clues from the blood, giving us a window into the genetics of a tumor without ever having to perform an invasive procedure.
So what kind of clues are we searching for in this blood sample? Well, we can look for whole circulating tumor cells that have broken off from the main tumor. We can find tiny fragments of circulating tumor DNA, which we can then sequence to identify the cancer's specific mutations. We can even detect chemical tags on that DNA, what we call methylation patterns, which can act like a return address, telling us not only that a patient has cancer, but potentially where in the body it came from.
And this brings us to our final thought for the day. I want you to really think about this. What will medicine look like when we can detect cancer at its earliest stage? When we can monitor in real time how a patient is responding to treatment, and when we can select the perfect drug for their specific tumor, all from a single vial of blood. This isn't science fiction anymore. This is the future that is happening right now as we speak. All right, that's all for today. Thank you.